Hi everyone, my name is Ed Davis. I am Senior Application Scientist for GeneCopoeia in Rockville, Maryland, and I'd like to thank you all for attending our webinar today entitled Functional Tags for ORF Clones in Your Research. We really appreciate it. Before I get started, I need to take care of a few practical matters. First, you may ask me questions at any time. However, you will not be able to speak vocally to me because your microphone is muted. Instead, please type any questions you may have in the question box in your control panel. If you cannot see your control panel, it might be hidden. In that case, look for a strip of buttons, vertical buttons, on either the left-hand or right-hand side of your screen, and one of these buttons will be orange with a white arrow pointing in one direction. If you click on that arrow, then you will be able to see your control panel. Also, in the interest of time, while I will take questions at any time, I will wait until the end of the talk to answer your questions, at which time I will answer each question in the order it was received. Okay, so now let's get started. Okay, so here are the goals of this presentation. First, I'm going to tell you how to use expression-ready ORF clones, a specialty of GeneCopoeia, in your research. And then I'm going to tell you how to choose tagged clones. And finally, I want to educate you on how to order expression-ready ORF clones from GeneCopoeia. All right, so here's an outline of today's seminar. First, I'm going to give you a brief introduction to GeneCopoeia and who we are. Then I'm going to tell you about our Omics Link brand of expression-ready clones. Third, I'm going to go in some applications for ORF clones. And then finally, I'm going to tell you how to order GeneCopoeia ORF clones and to show you how easy and powerful our ordering process is. All right, so let's start off with part one, Introduction to GeneCopoeia. Okay, so who are we and what do we do? Well, first of all, we are an original manufacturer and seller of a variety of research reagents and tools. And secondly, we're a biological research service provider to worldwide customers. So we have a vast product line at GeneCopoeia for products for functional genomics. And the first one, which is going to be the main focus of this talk, is ORF clones. And this is how GeneCopoeia was founded, on this product line and what we're most known for. But since then, we've added a number of interesting product lines that are usually related in one way or another to ORF clones. These are promoter clones, microRNA target clones, microRNA inhibitor clones, precursor microRNA clones, shRNA clones, and then we have a number of genome editing tools for TALENs and CRISPRs, and finally we have qPCR arrays. All right, so now let's move into part two, where I'm going to tell you about our Omics Link expression-ready clones. All right, so in the wake of the sequencing of the human genome, it became clear that once we had the genome sequenced, that much more work was going to be required for functional studies, mainly from identifying genes and how the genes are spliced up to the proteins that they encode. And so this really opened up a lot of avenues for research, because basically, when you look at something resembling the central dogma where DNA makes RNA makes proteins, we can see that the Human Genome Project was only, as big as it was only the first part of that dogma, okay, the DNA part. However, what happens with the rest of it? Well, naturally you make mRNA, and mRNA makes protein, so in order to do this, you need clones and you need ORFs in order to analyze various aspects of their biology, from either studying the regulation of the gene expression or by studying phenotypic effects of protein overexpression or mutated proteins being expressed, etc. All right, so the main focus of this talk is ORF clones. Well, why should you use ORF clones? Well, there are several good reasons listed here. The first is you can carry out biochemical studies in vitro, and most of these are based on macromolecular interactions such as protein with protein or protein with DNA, etc. Secondly, ORF clones allow you to carry out biological function studies in vivo, so you can study the effect of a protein overexpression, for example, in mice. The third reason is you can carry out orthologous studies. So if you wanted to express a human protein in mouse, for example, or a C. elegans protein in human cultured cells, having ORF clones allows you to do that with ease. Another big reason for using ORF clones is that it allows you to reverse or rescue a mutant phenotype. So you might have a point mutation in a particular gene of interest that causes a phenotype, and if you introduce back the wild type ORF back into the system, you can see if it rescues the phenotype. And last but not least, and certainly there are many others, but these ORF clones allow you to do drug target validations. All right, so GeneCopoeia, again, as I mentioned, was originally founded on and is best known for our full-length ORF clones. And in fact, we are the only company with a genome-wide set of untagged ORF clones. We also have a wide choice of numerous functional tags, which again is what the title of the seminar is focused on. And finally, and this is extremely important, all ORFs in our clones are sequence verified and guaranteed. And so the emphasis is that our ORFs are very high quality. All right, well, so what's so special about sequence validation? Some companies give you expression validation where you can see each ORF functionally validated for expression in a cell culture model system. Well, in those cases, those expression-validated ORFs are done with ORFs containing a tag like a flag tag, which allows you to conveniently detect the expression. But that's not always the best way to validate your ORFs, because if you don't sequence that ORF, well, then you could be expressing some kind of mutant with say a missense mutation, and the expression validation on a Western won't tell you that. Also, what an expression validation will not tell you is that the tag itself could be, in some cases, causing you to express some protein has either lost its activity or has acquired a different activity. But but what we can say is that if you validate by based on the DNA sequence, you know that that ORF is going to be expressed in the correct protein, whether it's tagged or not. All right, so now I'm going to tell you about the OmicsLink brand of expression-ready clones. Basically, we start out with a full-length cDNA that contains both the 5' and the 3' UTR, and again, this is sequenced. And then we take these cDNAs and we send them down 3 possible paths or avenues. The first is that we can tag them at either the 5' or the 3' end, and we also make untagged OmicsLink brand ORF clones. So here's a list of the advantages of the OmicsLink clones. First, they are ready for immediate in vivo and in vitro expression. So when you order one of these clones, you can drop it into your cells the very same day. Okay? No cloning steps are required. You don't need to do any subcloning. We also have the full-length ORFs without the 5' and 3' UTRs, so they are driven by heterologous promoters, usually the CMV promoter, but there are others. We also have a huge collection of ORFs. We have more than 45,000 ORFs from human and mouse. We also have these ORFs are available for human and mouse and even zebrafish, and they're available in lentiviral or non-viral vectors. So here's a list of some applications and advantages of the Omics-Link brand of expression-ready clones. First, they allow for high throughput and large-scale protein production and purification. Secondly, they're ready for various functional assays, including protein immobilization, cellular localization, and other functional assays. Third, they are suitable for reverse transfection arrays and nucleic acid programmable arrays, also known as NAPA. And also, you get a great savings in time and resources. All right, so our ORF collection, as I mentioned, is very extensive. The human collection of ORFs includes more than 21,000 ORFs, which again are fully sequenced. The mouse ORF collection contains ORFs for 15,000 genes, 13,000 of which are unique. And we also have ORFs from other species, such as zebrafish. Our our ORF clone, ready-to-express ORF clone systems also come with a number of promoters, fusion tags, and markers. So as far as promoters go, these are usually constitutive overexpressed promoters, although we do have some inducible ones. But the most common promoters are the CMV and EF1 alpha promoters, and we also have CMV and EF1 alpha in lentiviral vectors. We also have the T7 promoter for in vitro transcription or transcription in bacteria, and then some other specialized promoters like GAL1 for yeast-2 hybrid studies. In addition, as I mentioned, we have a wide variety of fusion tags. Some of these are for protein purification, mainly like His and hexahistidine and FLAG. We also have some tags that are intended for solubilization, like GST, maltose binding protein, and SUMO. In addition, we have classical fluorescence tags like GFP, RFP, and YFP. And we also have some good immunoprecipitation tags like 3XHA and CMYC. In addition, we have some more specialized and higher technology tags like Halo tag and Abby tag, and I'm going to tell you more about those later in the seminar. And finally, we have a wide variety of expression markers depending on your needs, most of the common ones such as puromycin, neomycin, etc. In addition, we focus heavily on what we term clone sets, where you can order our clones not just one at a time, but in various sets, and these can be either genome-wide or in various families that are either functionally related in some way or they could be part of pathways. So for the sets, the clone sets, these are CMV-based mammalian expression-ready clone sets, and they come either untagged, which can be advantageous because there's no potential for tag effects. They can also come with C-terminal flag tags or C-terminal monomeric EGFP tags. In addition, we have lentiviral clone sets, which again carry the CMV promoter, and these come either untagged or with C-terminal V5 tags. All right, so now I'm going to move into part 3, applications for ORF clones. So what I'm going to start out with, even though I mentioned in the beginning of the seminar that there's going to be a big focus on tag clones, we also want to emphasize our ready collection of untagged ORF clones. So why would you choose to not tag? Well, there are a number of good biological reasons, such as the tag could interfere with normal protein folding. Second, the tag could interfere with normal protein activity, even if it was folded correctly. Third, the tag could interfere with normal protein localization. Fourth, the tag could create a toxic fusion protein. Or finally, and certainly these are not the only reasons, but finally on this list, you might have a need for an antibody, but you have a native antibody, and so you don't need an anti-tag antibody. All right, so the untagged clones come in a variety of expression vectors. These are the most common. We have two types, both non-viral and lentiviral, and that's true of almost all of our expression clones. And these are usually just the ORF with no 5' or 3' UTR, and it's driven by a constitutive strong promoter. Most common is the CNV promoter in both cases, and they come with mammalian expression markers such as neomycin or puromycin, and of course bacterial markers. So GeneCopoeia, again, prides itself on the fact that we're the only company with a genome-wide collection of untagged ORF clones, and these have been used by a number of different research groups. And here is one example of a paper published last year by Al-Zubi et al., where they used a GeneCopoeia omics-linked clone to demonstrate that TNF-alpha overexpression eliminates breast cancer growth in vivo. So in this case, they showed that compared with the lentiviral control, there was no increase in tumor volume over a 30-day period. There was essentially no tumor formation. And the same is true of the tumor weight. The lentiviral control grows to nearly 200 milligrams, whereas there's no detection of tumors in the cells containing the GeneCopoeia omics-linked TNF-alpha expression clone. Another application of ORF clones is that you can validate RNA silencing. So you can put one of our full-length protein-coding ORFs into your cell line that's already had silencing against a particular gene done by double-stranded RNA or shRNA. And when you put that clone into your cell lines, you can see that it may knock down, or suppress or reverse the double-stranded RNA-mediated knockdown. All right, so now I'm going to go into the use of tags. So why would you use a tag? Well, one reason is that there's no native antibody available, so you might need to do an immunoprecipitation or a Western. And so if you have no native antibody, well, then you're going to need to detect it somehow, so a tag will help you do that. Secondly, tags can help you enable purification. Third, some tags can enhance solubility during purification, so some proteins are notoriously insoluble when expressed in E. coli, but addition of a particular tag might actually cause that protein to be soluble. Fourth, you might want to use a tag to study interactions with other macromolecules, such as other proteins or nucleic acids. And finally, some tags are good for doing subcellular localization or live cell visualization. All right, so what are some of the applications and advantages of the different fusion tags? Well, shown here is a list of the different tags that we have available for our expression clones. Some tags, such as His and flag especially, are great for purification, but they're not as good for other types of applications. However, some of our other tags are kind of the opposite, like GFP example is really not so great for purification or these other applications, but it's great for cellular labeling. Further, GST and maltose binding protein are ideal for increasing solubility, as well as SUMO. So again, with a large variety of fusion tags, you can determine what your needs are and then pick the appropriate tag. All right, so when one picks a tag, usually you have the choice of putting the tag at either the N-terminus or the C-terminus of the protein. Well, so that creates a choice, though, where you have to consider several factors for trying to decide whether to put the tag at the N or the C-terminus. These are the two main considerations. First, you need to choose an end that is not buried in the protein core, so it would be very helpful to have some structural information on your protein. And secondly, you want to choose an end that does not disrupt localization. So for example, if you have a protein that's localized to the endoplasmic reticulum, well, you might not want to use an N-terminal tag because that would mask the signal the ER signal peptide, which is usually at the N-terminus. All right, so now I'm going to go into some applications for tagged ORF clones. The first I'm going to discuss is protein purification, and one of the classic tags for that is the his tag, which is actually a hexahistidine tag. And the great thing about the his tag is it permits the use of classic metal columns or beads for purification, and it can be fused at either the N or the C terminus. We have a large number of vectors that contain hexahistidine tags, which can be expressed in either mammalian cells or E. coli. Shown here are two typical vectors containing the his tag at either the the N terminus or the C terminus, and we simply have the ORF cloned so that it creates a fusion protein with the tag, an in-frame fusion. So when you have one of these clones, you would then put it into your cells, and in the case of his tag, this can be mammalian cells, bacteria, cell-free systems, insect cells, etc., many systems. And once you express this clone in your cells, you then isolate proteins, and then you run your protein mixture over an affinity, either an affinity column, such as shown here, and in the case of his tag, it's either a nickel column or a cobalt column, or this can be done with beads, which can be either agarose or magnetic. And then once you bind to the column or the beads, you then elute your protein, your tag protein. Another great tag for protein purification is the flag tag. And flag is an 8 amino acid tag that is put at either the N or the C terminus. And the nice thing about the flag tag is it's a small tag that's less likely to interfere with protein function. And it's great for multiple applications such as protein purification, Western, immunoprecipitation, immunofluorescence, and again it can be placed at either the N or the C terminus. Just like the his tag vectors I showed you before, we have a number of ready-to-go expression vectors for the flag tag, and again these vectors fuse the ORF of interest in frame, either at the N terminus or the C terminus. Once that is done, once you have your flag tagged ORF in the ready-to-go expression vector, you then put the plasmid into your cell system, and again this can be many different types of systems. You then lyse your cells, and once again you can put your proteins into an affinity system, in this case a column or a bead with an anti-flag antibody, and then you can have your purified protein. Now I'm going to talk about live cell imaging, and one of the classic tags for live cell imaging is the GFP tag. GFP, as you may know, is a classic fluorescent marker from jellyfish. And it's actually a multifunctional tag. It's usable for live cell imaging, but it can also, in certain instances, be used for purification, Western, and IP. And again, the GFP tag can be fused at either the N or the C terminus. As I mentioned before, we have a number of ready-to-go expression clones for GFP, where the ORF is fused in frame to either the N or the C terminus. And this plasmid is then put into the cell system, but instead of lysing the cells, you then view the cells under a microscope. We also have another great tag in our ORF system for live cell imaging, which is known as the halo tag, which is from Promega. And, excuse me, I've lost control of my mouse here. Okay, so what is the halo tag? Well, the halo tag is a genetically engineered derivative of the dehalogenase, and it's a multifunctional tag. It's great for live cell imaging, purification, Western, IP, and more. And it efficiently forms a covalent bond with various synthetic halotag ligands, which help you with your different applications. And it's very versatile. It can be fused at either the N or the C terminal. So this is the way halotag works. if you have a halo tag on your protein, you then do a reaction with various different affinity ligands, and the available affinity ligands can have different fluorophores on them. So you can actually transfect or put these labeled proteins into your cells, and depending on the fluorophore, you can see different colors. And that exact example is shown here. This is data from our research and development department, where we transfected HEC293 cells with proteins containing the halo tag, but they're coupled with different ligands that fluoresce either red, green, or blue. And it's the same protein with the same tag. All right, so now I'm going to talk about another application known as pull down, which is similar to immunoprecipitation, except you're not using an antibody. But the whole idea is for you to precipitate proteins out of solution, which can either bring the protein down by itself, or it can help you to identify other members of a protein complex that physically interact with your protein of interest. And so one of the great specialized tags for this is the AVI tag. And so the AVI tag has several terrific features. One is that it gives specific and reversible binding of avidin or streptavidin to biotin. The tag can be biotinylated in vitro or in vivo by biotin ligase. The tag can be fused at either the N or the C termini. Biotinylation can occur in vivo or in vitro, and this is a very small tag that's less likely to interfere with protein function. As with our other ready-to-go expression clones, we have ready-to-go expression clones for AVI tag, where the ORF is fused in frame either at the N or the C termini. And in this case, one introduces the plasmid into the cell system of choice, and now the cells are lysed and the proteins are isolated for either Western or mass spectrometry for identification. Right, so now I'm going to go into the final section of the talk, and that is to show you how to order GeneCopoeia ORF clones. So what I'm going to do here is actually, this is a diagram of our web page, but what I'm going to do is I'm going to jump out of the PowerPoint and do a live demonstration for you. So let's escape from the PowerPoint and go to a web browser. Okay? And I'm going to open up our home page. So the goal of what I want to do here is to perhaps order a tagged ORF clone. And so I'm going to search for SOX2, which is a sex-determining factor that was originally identified in lower mammals, but it's also in humans. And so I'm sorry, I'm actually not going to use the simple search. I'm going to go to our advanced search page. So the advantages of our advanced search page is that they give you many search options. First of all, you can restrict your search to various species, such as human, mouse, and zebrafish. You can also search by restricting to various gene categories like gene symbols or pathways. Or again, you can restrict. You can restrict to 2 species at once if you want. You definitely have a lot of options. So what I want to do is I want to find a human SOX2 clone. Okay, so I selected human and I'm going to type in SOX2. Okay, and then press the search button. So what comes up is the human SOX2 gene ID. Now, this is basically a portal to many different clones that contain SOX2. Okay, and to go through that portal, I'm going to click on the product ID shown here. And when I do that, I then get many options for ORF clones with SOX2. Now remember, it's the same SOX2 ORF, so it has the same product ID as you can see here, but it has many other options which are based on either the marker or the tag. So I'm interested in ordering SOX2 with an N-terminal GFP tag. So I find that here, and if I want to, I can look at the map, look at a basic plasmid map, and when I do that, I get the general plasmid map similar to what I showed you before, which shows an ORF tag with a N-terminal GFP. And I can see that it has a neomycin marker and it's driven by the CMV promoter. Okay, so I'm going to exit that. And so that looks good to me, so I'm going to select that clone, that plasmid. Okay, and then I'm going to add this to a shopping cart. Now, so this pops up briefly. What you saw was a brief pop-up where it tells you what kind of plasmid is delivered. And in this case, it's in DNA. So when this happens, now I want to view the shopping cart, and if you haven't registered, then you need to register an account with us. But I already have an account, so I'm going to log in. Okay. And there's my shopping cart. Okay, so this is purified plasmid, and we have many different price ranges, but the price for this particular clone, this GFP tagged SOX2 clone, the regular price is $805. However, we are running a special, a 30% off special on all of our ORF clones until the end of February. And so in this case, this particular clone costs $564. Okay, now I just realized something. I thought, I think I really want to have the untagged version of this because, as I mentioned before, sometimes tags can interfere with functions. So having an untagged version would be really useful. Okay, so all I have to do then is just leave the shopping cart, and this clone will stay in the shopping cart, and I click on back to product page. So that brings up the same list of clones for SOX2 as I saw before. So what I want to find is the equivalent clone that has CMV promoter and neomycin marker but no tag. And so that's shown here. It's our M02 series, CMV promoter, neomycin marker, and no tag. So I'm going to click on that, and I'm going to add that to my shopping cart. And so now this window pops up, and I have several different options for this particular clone. And I see that the first option is bacterial stock with next day shipment. So I think, well, that's great. I think I'll go with that. That way I don't have to put the plasmid DNA into bacteria for this particular clone. So I'm going to add that to a shopping cart. And that should bring, that brings me up to my shopping cart. So again, this particular clone is actually one of our budget-friendly clones that I'm going to talk about in a little while. So it is actually vastly reduced in price, down to $175. And so now this is my shopping cart, and I decide, okay, this looks great, and then I will go ahead and check out, which a process I'm not going to go through now. So I'm actually going to go back now to the PowerPoint. And I had some demo slides just in case there was a technical issue. So I'm going to skip ahead from those. And, here's something else I wanted to tell you. Not only can you order directly from our website, but we also have a link to give you the ability to order from NCBI, shown here. So I'm going to once again exit the PowerPoint and go back to my web browser. And here I am on the NCBI website. It's set for PubMed, but what I'm going to do is I'm going to search the gene database for SOX2. Okay, so I type in SOX2 in the search field, and I press search. And what I see here is that I see the human SOX2, which is what I was interested in. So I click on that, okay, and actually I click on the name, and what comes up is the information for SOX2. Now what you'll see over on the right-hand side among different lists under related information is order cDNA clones. I think, great, let's order a cDNA clone. So when I click on that, I now see several vendors from which I can order the cDNA clone, and one of which is us here at GeneCopoeia. You can ignore the other vendors. So then anyway, you just press on this order button and it would take you to our home page to order the clone, which a process again I'm not going to go through. All right, so let's go back now to the PowerPoint. And let's move on to the next slide. So now I'm going to tell you a little bit about our ready-to-express ORF clone pricing. Most viral and non-viral clones start at $805, and the prices can vary depending on different factors, such as larger clones simply cost more because they require more work to deal with and more gene synthesis. But the starting price is usually around $805 for most clones. However, I mentioned we have a 30% off promotional price through the end of this month, so now they are starting at $564. We also have a type of clone, which I'm going to go into in a little while, called budget-friendly. These usually start at $250, but right now, again, we're running a promotion where they start at $199. And I also forgot to mention earlier that we have secondary clones. In the case of the SOX2 search that I did, the second clone I put in was much cheaper. It was only $175, and that's because it was subject to our secondary clone discount. What secondary clone discount means is if it has the same product ID but a different tag, well, by ordering that second one, you get a discount. And it's usually around $200, but again, they start at $175 till the end of the month. So that's the secondary clone discount. All right, so I also mentioned the budget-friendly expression clones. So one question is, what are the budget-friendly expression clones? Well, budget-friendly expression clones are a set of more than 20,000 human ORF clones, many of which, though not all, are ready for next-day shipping. In addition, these often come with C-terminal flag tags for the non-viral clones, or they come untagged, and they come with the CMV promoter, and they come either in non-viral or lentiviral vectors. So the budget-friendly clones are great for saving you lots of time in the case of the ready for next day shipping clones, and lots of money. All right, so I also mentioned earlier in the talk that we have clone sets. These can be genome-wide or in families, and I went through these details earlier. The non-viral vectors come either untagged or with C-terminal flag tags or C-terminal monomeric GFP tags, whereas the lentiviral versions come either untagged or with C-terminal V5 tags. So the clone sets can come either with genome-wide coverage, where you can order whole genome mammalian expression clone sets with no tags, and we have more than 12,800 of those, of genes, more than 16,000 clones. And again, they come either in non-viral or lentiviral vectors. We also have a number of gene sets that come in gateway vectors, which I will mention later. We also have clone sets based on gene families. So we already have a number of set design families, such as CDs, cytokine receptors, drug target genes. There's a wide selection to choose from. And the features of these is that they're non-viral. They're either non-viral or lentiviral, and they come expression ready, as I mentioned before, either untagged or with C-terminal flag tags. The lentiviral expressions are native expression only. Some come as gateway plus shuttle vectors with no promoter, and we can even make custom sets for you. Alright, so now that I've told you all about our Omics-Link brand of expression clones, I'm going to tell you briefly about other GeneCopoeia ORF clone products. The first one I'm going to briefly mention is our Safe Harbor Integration ORF clone collection. GeneCopoeia is also known for our genome editing tools such as TALEN and CRISPR. And so we have a large collection of clones, over 18,000 sequence-verified human ORFs, which are designed for knock-in at the so-called safe harbor loci at either the AAVS1 site on human chromosome 19 or the ROSA 26 allele in mouse. Actually, these are human clones, so they're human only. So we can use either use TALEN or CRISPR to knock in these ORFs with various markers and promoters into the AAVS1 site. Also, we have another product line known as the Overexpress Gateway Plus clones. These are not ready to express. However, they have a number of great features where they carry both recombination and multiple cloning sites, so they can be readily moved into other vectors by either the convenient gateway cloning system or by conventional restriction enzyme mediated cloning. We have more than 20,000 human and more than 15,000 mouse clones in the Overexpress Gateway Plus system. And these allow for simple and rapid transfer of protein coding ORFs into any gateway destination expression vector. They are available with and without stop codons, and they contain ribosome binding sites, either Shine, Delgarno, or Kozak, for optimal translational context. Alright, so now I'm near the end of the talk, and so I just want to tell you, I talked primarily about ORF clones today, but I also mentioned our other product lines such as lentivirus, stable cell lines, and shRNA. And the reason I'm showing this figure is to show you that these other products are all related in one way or another to our ORF clone product line. Again, you can get ORF clones in either non-viral or lentiviral vectors, and we have packaging kits to help you do an entire experiment with GeneCopoeia clones. We also have stable cell line services where we can overexpress a particular clone of interest in a particular cell line for you. We have shRNA clones, which can be used in validation studies of either the shRNA or the ORF. We have qPCR reagents and arrays, which can be used with our ORF clones, and we can also use our ORF clones to express recombinant proteins for you. Alright, so with that, now I'm at the end of the talk, and I just want to briefly summarize. So first I told you about the GeneCopoeia Omics-Link brand of expression clones. These are full-length ORF clones for human, mouse, and other systems, and they come ready to express without any tedious cloning or verification steps. Secondly, I told you that the GeneCopoeia Omics-Link ready-to-expression clones come either untagged or with a large variety of functional tags for virtually any application. Third, I told you that GeneCopoeia is the only provider of genome-wide untagged expression-ready ORF clones, and that all expression-ready ORF clones are sequence-verified and guaranteed. I also told you that we have a number of protein fusion tags that can greatly facilitate your goals of detecting, purifying, and localizing your protein of interest. And I also showed you that ordering Omics-Link brand ready-to-express clones from GeneCopoeia is as easy and convenient as the click of a mouse. Okay, so that's all I have to tell you about today. I just want to thank you again for attending the presentation, and if you have any additional questions, please call at 1-866-360-9531 or visit our website at www.genecopoeia.com. And now I would like to take questions. So if you have any questions, please, as I mentioned before, type your questions into the questions field. Okay, are there any questions? So far I don't see any. All right, does anyone have any questions? Now is the time. All right, well, I don't see anybody asking any questions, so with that, I'm going to end the presentation, and once again, I'd like to thank you all for attending. I really appreciate it. And with that, I'd like to wish you good luck in your experiments and to have a great day. Have a good day. Bye.